So good morning all. So we are going ahead with the next topic, and as I told you yesterday, we have planned in such a way that all the topics to be covered in this lectures, and basically that is related with the cancers. So as we were planning, we found that there are certain issues, maybe the topics. Dr. Nag, he is actually covering almost all the topics related with the cancer. So he is telling you about the advanced techniques. He is telling you about even the maybe the models what he is using, as well as the advanced cancer models, and along with that even he is telling you about even the clinical trials and all these details. So what I found was that in between maybe we can include certain things where we can focus on the natural products. Even he will be telling about the natural products also, but his goal of, or maybe the way he is telling will be different, because his focus majorly on the drugs, what he is telling about the clinical trials and FDA drugs and all. So what I thought was that better let us go ahead with certain studies which is done at our center, and which can be linked to this topic, where we are actually trying to find out a natural compound, and that natural compound how it is involved in the prevention of cancer at the basic level, so that it can be linked. Because what I was going through, I was found that there was no mention about the isolation of the natural compounds. I thought that is a small study that can be presented, that fills the gap, one thing. And the other topic which I am going to tell you tomorrow is about how bioinformatics can be used to find out or maybe target the cancer. So these two aspects I thought he might not be covering. So let me maybe fill that the whatever the gaps that were there. And because he is covering almost everything, and he is expert in even in clinical trials. So this is something where we are actually using the drug or maybe the source which is indigenous to maybe our places, and that can be basically worked out in such a way that we can find out some few drugs which can be taken further. Okay. And here my idea was that, because now even the Indian government, it is actually promoting about indigenous knowledge, or as well as Indian knowledge system, as well as the whatever we have the resources, that best way how we are utilizing. So that need to be explained. Maybe we are doing at the preliminary level, and that can be taken further for higher studies. That's what our intention was. And along with whatever Dr. Nagi is telling, maybe this is a basic study that may complement what he is telling about. Because the certain aspects, we do have certain limitations. We may not able to do maybe certain studies what it is done at the higher level, maybe the institute level. As a state university, we do have our own limitations, and maybe the funds or maybe the some other aspects. So with few collaborations and all, how we can establish a simple study, and we can perform the maybe the basic research that I am going to tell you now. And along with that, I need to mention about the Mangalore University. So we have a different departments basically, because those who are coming from outside, you might be not be familiar with what facilities and what type of subject experts we have. We do have chemical science and biological science, as well as computational science, and they are leading actually in their own field. And in chemical science, we are working on various compounds like indole compounds, or various maybe there are certain compounds which are having the capability to prevent this maybe the what do you call the cancer activity, or even other activities has been checked. And even there are certain compounds that have been patented in this Mangalore University. So along with that, even in computational studies also, they are doing wonderful job in maybe designing different softwares. And we had our own software. Yesterday I was mentioning you might have heard about Emulink software that was actually developed by our Honorable Vice Chancellor. At that time he was actually under his leadership it was developed. It was actually in-house the software. Along with that, even we do have other departments, they are having different collaborations, and recently we had applied for many more projects. Sorry, I forgot to mention about nuclear science department. There are many projects they are going on, where on the radiation studies they are publishing wonderful maybe the publications, and it is directly it can be implemented to the society. That kind of informations it is generated. We have the collaboration with BARC India, where with the collaboration many more studies they are going on, and even we do have the instruments, and because of that instrument many of our faculties they got funding from various agencies, and these are the basic aspects about the department as well as the Mangalore University. And just let me tell you brief about this one. Already Nag has explained various aspects about the cancer. I will go with the basics only. So cancer, as we know, it is one of the leading cause for the maybe the morbidity as well as mortality. And even in India, maybe the rate of this cancer is maybe increasing day by day. So according to the Globocon, it is 19.3 million new cancers cases and almost 10 million cancer deaths in 2020. So I have listed out few cancers where it is considered to be the leading cancer. So the one, maybe that is the first place, it is breast cancer or lung cancer, colorectal cancer, or even prostate cancers. They are considered to be the leading across the globe. And even cancer deaths, you find lung, colorectal, and liver and stomach. And you can observe the maybe whatever the cancers I am telling, it is something to do with the lifestyle of a person. Okay, whether it comes to maybe except breast cancer, very difficult to predict. Otherwise, the lung cancer or colorectal cancers or even liver cancer, most of the occasions, they are some way or the other, they are linked to maybe the lifestyles. There may be different causes also, as Nag was telling about the genetic causes and other epigenetic causes and all. So this is actually published in WHO website, and this tells about what is the scenario in India about the cancer. Okay, so there are nearly 1.4 million the cancer new cases and around 0.9 deaths, and that means it is it is maybe increasing in alarming state. So we need to focus on this work, and that's why even the government of India, it is actually supporting in this budget also they have included cancer is one of the major concern for the society. That's why even in the budget they have maybe they have included some amount so that maybe the screening as well as treatment can be done using the budget. So what are the top 5 cancers in India? If you go through the WHO website, so there are certain cancers, they are leading in case of males, and there are certain the cancers, they are in females. Males may basically lung cancer and lip cancer or mouth cancers, or maybe esophageal cancer. All these cancers, they are in the leading condition, whereas in females, even here also the breast cancer is in the highest, actually the incidence is actually breast cancer, followed by the maybe the other cancers. And in both male and female, if you observe, the cancer incidence with respect to mouth as well as lip is high. That means maybe the lifestyle is actually causing many, that is actually the one where the chewing the tobacco or something like that, that is actually the causing that maybe the mouth cancer or esophageal cancer and all. Maybe that's why even the government has banned gutka so that the cancer can be avoided. So just let us see briefly about what actually causes the cancer. So I have listed out only few. Maybe the factor will not limit here. There are other causes also to the maybe causes that causes cancer, even the hereditary or, and here the problem is that as it is a heterogeneous disease, there are n number of causes that causes the cancer. So physical carcinogens, as you know that UV radiations or any ionizing radiation that may cause the cancer, or even maybe in the case of this Mangalore, actually it is not advisable to move around at the noon time because it is a high temperature one thing, and it may lead to the kind of the maybe the health problems. And along with that, even there are chemical carcinogens like asbestos or components of tobacco, smoke, alcohol, aflatoxin. This aflatoxin you might have, maybe day to day we are using this one. Unknowingly or knowingly we are using aflatoxin with us because we are habituated with using groundnuts, and you might have observed that the powdery kind of thing on the groundnut, or even onions, if you peel it off, there is a black color, that fungi, it is growing on that one. That is actually, if you find that if it is covered completely with the fungi, that is the aspergillus, that may be one of the major reasons for the cancer. Because if you ask any person maybe who is suffering, he will not have any habits of either drinking alcohol, or they don't smoke, or they don't have any habits that will cause the cancer. But if you carefully observe, they will be using one or these things, like either they may be using the onions, maybe grown with this fungus which releases so many types of this aflatoxins. They are highly carcinogenic. Or even groundnuts for that matter, they cause actually the maybe they contain the aflatoxins. So along with that, even arsenic in drinking water, that is also a contaminant. Many places we can't have the potable water. So these contaminants can cause the cancer. And along with that, even the biological carcinogens, as Dr. Nag once again he mentioned about Helicobacter pylori. That one work actually we carried out in Mangalore University, interestingly. And here the people from nuclear science, medical physics, they carried out this study, where they found that they have designed a technique to find out the presence of this Helicobacter pylori by taking the breath of, or maybe the analyzing the breath of a person. And they found that in the northern part of the Karnataka, many people they had Helicobacter pylori, but they did not having any maybe the symptoms that is associated with the Helicobacter pylori. But we could not continue that study because of various reasons. But it is an interesting finding. Maybe it is showing that many people are actually having this Helicobacter pylori in their, or they are infected. Along with that, there may be other viruses, they might be causing the cancers. That's why usually the doctors, they advise to go for various checkups whenever they suspect some maybe the general symptoms of a person. And even bacteria, parasites, they may cause the cancer. So what are actually the important characteristic features of cancer? So I am going with the basics. So here this was actually started with Hanahan and Winberg. They proposed different hallmarks of the cancer, and these were considered to be the characteristic features of the cancer. If you look at the data given here, there are various factors they are actually responsible for the development of cancer. One is that maybe I will go with any one random way, that is either maybe sustained proliferative signaling. Usually we do have a proper signaling. We do have our own control. It is just like a society. Society has a definite order and definite hierarchy. Even in the cells, we do find the same discipline what we follow in the society, or a healthy society, what I am telling. But out of that, one or the other becomes the antisocial. Okay? If you don't control that antisocial, it may lead to the maybe the chain of events. It may lead to the maybe different activities that may increase the number of antisocial elements in the society. And in the same way, even in our body, there are so many the healthy cells will be there, or a functioning cells will be there, and all other cells will be there. And if they are not getting the proper signal, and if they are not controlled at their time, they may develop into the cancer. Or even that actually the signaling itself, that's what, because whatever I am referring, I should refer to Nag, because he has done practically all that work what I am mentioning. Okay. And then is that deregulation of cellular energetics, whatever regularly it happens, it may vary in the cancers, or evading growth suppressors. Usually what happens, the cells need some signals to grow also. And they do have, there may be some limitations in them once the growth factor stops. But what happens is that in the case of cancers, they are actually not responding to the, they may not depend on the growth signal, or it is a mutated growth signal where it is continuously actually making that cell to divide, and that leads to the cancer. And one more thing is that inducing angiogenesis. Angiogenesis is, in the sense, it is the formation of blood vessels. We do need blood vessel. When you have a wound, or when there are different stages of the growth, there is a need of the proper blood vessels. But what happens is that these blood vessels, they are actually controlled. Whenever they are needed, maybe in the case of the females, the endometrium, it is regularly damaged and it should be regenerated during the course of time. That is taken care by actually angiogenesis. There are different stages like angiogenesis and vasculogenesis. Vasculogenesis is a kind of the formation of the blood vessel de novo, whereas angiogenesis, it actually starts growing from the existing blood vessels. Okay. In the case of cancer, what happens, as the cells number increase, the mass of cells they increase, it makes that cells to crave for the nutrition as well as oxygen, because we are aerobic in nature. We need the oxygen as well as the nutrient for this, maybe the cells, they need the nutrients. In such condition, the blood vessel is the maybe the mediator providing all these nutrients as well as oxygen. In such case, if it is a normal blood vessel will be formed. But in the case of cancer, what happens, as there is a proliferation of cells to that much extent, the number of cells will be so high, it gives the signal to the blood vessel that we need more blood vessels. And these blood vessels may not be a normal blood vessel, they become a leaky blood vessels. And as a result, what happens, there are many blood vessels near that cancer tissue, but they may not be doing a proper function, or it may favor the growth of the cancer. And along with that, even alteration in the case of the activating invasion and metastasis, the tumor will not stay in that place. Wherever it started, it actually disturbs the layer on the maybe the blood vessels, and it may cross to the different regions, and we call that condition as metastasis. Suppose if there is a cancer in the maybe the liver, it can spread up to some other organ which is distant from the liver. So that we call it as metastasis. And even resisting cell death. Cell death is one of the key feature of all the normal cells. They have certain lifespan like that. Suppose if a person, if he doesn't die, he becomes Chiranjeevi, then it is not possible. Here, in the case of these cancers, they are continuously actually getting divided, and they are there is no control over that division. And along with that, enabling replicative immortality and genome instability is the one that actually the hallmarks of the cancer, where DNA is getting altered, and there are various alteration that takes place, and it may lead further complications, or even tumor promoting inflammations. That also one of the hallmarks of the cancer. And as Nag was mentioning, nowadays the trend is actually the immune cells and how they interact, because the immune cells are the one, they help to maintain the proper health. Suppose how they evade that immune cells to become cancer, that is actually considered to be the hallmarks of the cancers. And suppose if we are looking for the cancer therapy, what exactly we are looking for? So we are looking for, because if I am a lay person, I don't worry about what drug it is, whether it is a plant drug or animal drug or what drug it may be. It should increase my lifespan. That is the progression-free survival. The person should not have the cancer, and that cancer, if it is there also, it should not progress further, and that the patient will be looking for. Or overall survival, how far extent after taking the drug the patient can survive. So if the patient can survive only few days, it may not be beneficial for the patient, because he will be spending lot of money on getting the treatment. And in a country like us, we have a mixed population where we have poor as well as rich, and in between the middle class. Any disease, especially the cancer, will be burden on the family. So what way we can increase the overall survival of a person, that actually matters a lot when we are looking for the cancer therapy. And later, what is the safety and quality of life he will be leading? He may survive, but what quality of life he is surviving is also one of the major questions that should be answered. And biomarker response. So later, once it is done, that is, we usually do all these things in the in vivo models. Then we will try to find out what is the mechanism of action of that particular drug, or how is there any resistance for that drug, or maybe if there is any novel biomarker, can we identify that one? And later we will try to ask a question whether intervention improves outcomes compared to the standard care, already what we have. That is actually the goal of the cancer therapy. So we have a different variety of cancer treatments. So basically the cancer, if you look at the history, the cancer history starts with most of the occasion. It starts with the surgery, because when the cancer, maybe once it was actually studied, or the doctors, or when the patients used to go to the doctors used to think that the removal of the mass is the best way. That's why the surgeon who was good at removing the tissue from depth was considered to be the best surgeon in that time. Maybe in 1930s I am telling about. But later on they found that due to the surgery, there are other organs are also, they were getting damaged. And as such, especially the breast cancer was studied in detail at that time. And later on, because there was an invention of this radiations and all, radiotherapy started. Along with that, even the chemotherapy started. Till now we are using these three as the conventional anti-cancer therapies. Along with that, even nowadays there is a maybe advances in various types of the cancer therapies like hormonal therapy, or even bone marrow transplant, or even the immunotherapy. That's what Dr. Nag he was telling about. Nowadays everyone is moving towards the immune system and how best it can be targeted in such a way that the cancer can be cured. And maybe the last one, what I am telling about is that personalized therapy. So where the drug which is acting on one person may not work the same way on the other. Suppose if you take maybe any tablet, someone may get cured within maybe 1 hour or 2 hours, or someone may take around one day or something like that. It is just like a cold. The person who is sitting next may not have cold also. But maybe there are some people, even by just if anyone else sneeze, they will catch cold. So that depends on person to person. We have our own microenvironment in our body, just like the way we think. Okay, so that's why nowadays even the personalized therapy is getting momentum. And whenever we go for the preclinical studies in drug discovery, so what we are actually looking for? So we are looking for we are actually carrying out various studies like in vitro study, in vivo study, or ex vivo study. Even sometimes we need in silico studies. Okay. So whenever we are going for the preclinical studies in drug discovery, the best way what I feel is that target identification. Maybe we are going to the lab, maybe spending so much work on the lab. Better can we work in the simulated condition in the computer and find out the target, so that working on the animal or working on the drug will be much easier. And once you are proved that, or you are confirmed that the drug will act on that particular maybe the target, then you can move ahead with the maybe in vitro studies, as well as you can go ahead with the mechanism where exactly it binds, whether it binds to certain enzymes or whether it binds to certain receptors. All those things can be studied. And in the same time, even we are supposed to find out, or we have to validate the target. And it might be either genetic, molecular, or pharmacological tools we are using. And we have to optimize and formulate the drug. Maybe sometimes one drug may not act completely. That's why we are now have a drug, the maybe combination of drug that may work out. That's why if you go to maybe if anyone has visited the hospitals, if there is a cancer patient, they will not prescribe one drug. Always the patients, they will complain that we are given a list of drugs. But the drugs are given in such a way that one should complement with the other, so that the person should be healthy. And ultimately we are supposed to find out the mechanism before going to the in vivo or maybe the clinical trials. So far whatever we are doing in the animals and the cells, that should be carried to the human beings. Before that, you should know how exactly that drug it acts, where exactly it is acting, what is the signaling pathway that is actually getting targeted by this drug. Once you know that one, then it is easy to plan the clinical trials. So why? Because my talk is basically about the natural products in cancer, why the natural products are actually important and they are given a weightage. The major thing is that nearly 60% of the anti-cancer drugs, either they are derived or inspired from the compounds from either terrestrial plants or sometimes microorganisms or even marine organisms. So terrestrial plant based drugs, you might have heard about the famous drugs like Taxol, that is derived from barks of the Taxus brevifolia, or Vimlastin and Vincristine, that is from Catranthus roseus, Vinca rosea, or microorganism based drugs that includes doxorubicin or actinomycin D, or rapamycin, where the Nag will be telling about mTOR signaling. Initially it was the signal, that is actually the rapamycin was actually studied on yeast. They found the target, that is rapamycin target. Later they found the same thing in human. That's why that was called the mTOR and different TOR signals. And marine derived drugs, there are different drugs. They are actually derived from the marine organisms or even the sponges. They are helpful. And here, whenever I am mentioning the natural products, some of them they are in clinical trials. In detail, Nag will explain what are the drugs they are used in the clinical trials. And this is the chart I have taken from the Newman and Craig. They are actually they are working on this maybe since many years about the statistics of the natural products. And if you look at the graph where that green mark it shows that either they are the plant derived or they are actually kind of the one maybe natural product, botanical or derived from natural products, all those things. You find nearly 50 to 60% of the drugs, they are actually derived from the this natural products. That is natural product, what I say, it includes the biological, maybe the proteins or whatever it might be that is isolated, or including the herbals, or maybe other compounds which is isolated, all those things. The synthetic actually, they are having, maybe you can see that in the top you find there are certain the synthetic one. And if you look at the statistics from 1981 to 2019, you find that there are various drugs, they are actually studied, and prominent one is actually the natural one, or maybe the botanical based drug, or maybe the mimics of the natural compounds. So there are certain, basically I am taking only few that natural compounds which can be considered, maybe they are actually studied in details, and there are certain anti-cancer compounds, they are in the clinical trials also. They are some of the drugs, they are even FDA approved for the use. And alkaloids, as you know, most of you, maybe some of you are chemistry background, and you know that they are nitrogen containing compounds. Most of the alkaloids, they have studied the mechanism of action. They found that these are the major mechanism by which the anti-cancer compound, these alkaloids, they are working on. So one is that the microtubule inhibition, so there won't be any cell division, or even there are certain topoisomerase inhibitors where topoisomerase is needed for the DNA replication, that you know, and if the replication is blocked, that may prevent the cancer progression. And even sometimes apoptosis induction, so that they induce the cell to go for death, so that the cancer cells can be controlled. And metabolic modulations, there are certain modulators, they will alter the way maybe the metabolism that takes place. And along with that, even anti-angiogenesis and metastasis suppression, that is actually promoted by these alkaloids. These are the key mechanism that has been studied with respect to alkaloid anti-cancer drug, like Vinca alkaloids, or maybe camptothecin derivatives, podophylla toxin derivatives like etoposide or teniposides, or even other approved alkaloids, what I have mentioned here, like homoheringitonin. All these alkaloids, they have a definite mechanism on acting on the cancers. So these are the few anti-cancer drugs I have included. So this is Actually, I have not included an exclusive list of all the anti-cancer drugs, just to show you that there are certain compounds, they are acting as cancer. And next thing is that flavonoids and phenols, they are actually polyphenolic compounds. They usually fruits, they are considered to be rich in these polyphenols. They have the mechanism of action like induction of apoptosis or inhibition of angiogenesis, similar in that way, or cell cycle arrest, or suppression of oxidative stress and inflammation, inhibition of metastasis invasions, all those things. And when I say the flavonoids, usually it comes to our mind is that curcumin. That's why Dr. Nag, he was telling about what is the problem with the turmeric. Usually it is it has less bioavailability. That's what I was actually maybe going through a paper. There is a controversial statement about the curcumin. There are some paper, they claim that along with that there are certain chemical, the bioavailability can be enhanced. Like usually we in Indian system, we have a habit of using curcumin along with the pepper. So some studies, they have shown that curcumin with the piperine, actually its bioavailability is actually getting increased, but some papers, maybe some papers, they deny that actually the claims. So it is slightly controversial, and the same thing even it can be told about resveratrol, that is from grapes. It was considered to be the actually anti-cancer. That's why the red wine is considered to be the very healthy and all. That claim, it is a controversial claim. That's what some paper they say. And even ellagic acid, that is derived from pomegranate, that is actually an maybe flavonoid, which maybe Nag, he will be telling in detail about that one, derivatives of the ellagic acid, which how it prevents the cancer. So not only these two, the major category is that even terpenoids and steroids, they are used in the cancer therapy. They include, and basically terpenoids and steroids, they have slight difference. So terpenoids, they are derived from isoprene unit, and even the steroids are also derived from the isoprene unit, but there is a slight difference where the steroids are the 4-ring carbon containing structures. If you look at the reported literature about these steroids, you find that they act in a different way. So like induced apoptosis, or they may target oncogenic pathways, and even reduce angiogenesis and metastasis, and they may act as they may have anti-inflammatory effect. That's why whenever the inflammation is high, we are going for some steroids, so that maybe the pain or something, it can be reduced. So like dexamethasone or prednisone or methyl prednisolone, hydrocortisone, and all these things, there are some key drugs that have been reported to act as anti-cancer in nature. So what are the advantages of natural products in cancer research? Why the people are looking for the nature to find out the drug for what we have in the nature? Okay, the best thing is that the low toxicity, and the another one is that the beauty of this natural product is that they have synergetic effect. Okay, sometimes they may be used along with certain other therapies also. Like maybe some paper, they claim curcumin actually it enhances or it sensitizes the cancers when it is combined with other chemotherapy and radiotherapy. Or another way is that there are certain plant products or maybe natural products, they are very good immunomodulators. That's why even when the COVID was there, the government has suggested to go for certain immunomodulators, like maybe there are certain herbal combinations, so that the modulation actually makes our immunity strong, so that you will able to face that the cancer, so that they stimulate the immune system and helping the body to fight the cancer. And along with that, if you look at this, the natural products or natural compounds, these compounds, they not act at one signaling pathway. They may be acting at different signaling pathway. So just I have taken one example of curcumin, where it is showing it is involved in so many different pathways. So it actually gives us the maybe the information that they may have a complex nature of acting on our body. So next, let me come maybe discuss about what actually we are studying. I am actually focusing on endophytic fungi, which is maybe one of the microorganisms, and there are various definitions about the endophytic fungi. So these fungi, there are different types. This endophytic fungi is the one which is present within the plant, and there are different theories about these plants. So they are associated symbiotically, and they provide maybe there are different secondary metabolites to the plants, and in turn the plant will give shelter to the plants. And this is the definition I have taken from Venn et al. It says that fungi that live in plant tissue throughout the entire or partial life cycle, and establishing beneficial symbiotic relationship. And if you look at the plants, there are around 300,000 terrestrial plants. Almost all the plants, they harbor either one or the other this endophytic fungi, indicating that if all the plants they have that many number of endophytic fungi, there is a close relationship between those two. And whether any work has been worked on the endophytic fungi. So if you see the literature, you find it has been worked on various studies, like whether anti-cancer, antimicrobial, anti-diabetic, and all those things. I was searching whether is there any FDA approved maybe a drug either for clinical trials or in the market. As far as my knowledge is concerned, no drug that is actually so far it has not gone to the clinical trials. So it is under the maybe the preclinical trial stage itself. There are many maybe the molecules, they are reported, and they are at the preclinical stage, because it is slightly difficult to work on the endophytic fungi. Now the major question is that why we have selected the endophytic fungi. And as we were going through the literature, we found that there are certain literature, they say that whatever the medicinal activity of the plant, it is because of the endophytic fungi. And the other way, there are certain reports, they are telling that plants are the one actually they are giving the maybe secondary metabolites to the endophytes. So whether it is the endophyte that is giving out the medicinal medicine, or the plant actually getting the medicine from the endophyte, is still it is under debate. I can give you example why these kinds of debates usually they arise. One is like that Taxol. It is produced from maybe the Taxus brevifolia, and at the same time the microorganism like fungi, they have the capability, like Fusarium, they have the capability to produce the Taxol, okay, which is present in that it was isolated from that plant. And the other one is that Campothecin. It is basically in plant, that is Campotheca acuminata. The same thing, it is actually produced by the fungi which is growing on that particular plant. So whether the plant is responsible for that active component or the endophytic fungi, we don't know. So that's why we were very much curious to study that one. And why we have studied endophytic fungal research. So does it have any implications? Because this is closely linked with the job. Because when I was posted, if you look at the first picture, it is actually FMKMC College at Madikeri, and later we shifted to a place called Chikkalavara at Kushalnagar. They are the remote place, and one thing what I was observing was that whatever you keep fungi used to grow. And it was easy to grow the fungi, and moreover we had limited resources, and best way to work on that or any work is that you work on the fungi. It is easy to carry out the study. So, and moreover, we had enough number of medicinal plants because it is at the Western Ghats. So many medicinal plants they were available, and we thought that why can't we work on the endophytic fungi? Because that time many people they were not actually working on that endophytic fungi. I am telling about nearly 15 years back. So we thought that let me concentrate or we can focus on endophytic fungi. One advantage is that growing is easy, and the material what they are required is less, and the contamination problem will be less compared to bacteria or maybe the plant components what we are using. So then we thought of selecting the plants, because when you work on the endophytic fungi, what plant you are supposed to select? My intention was very clear. If you send the research scholar to collect the plant from some place which is around 300 kilometers, he will go to his native place and he come back. By the time the research time is over. Why can't we select the plant which is very near to adjust into our place only? We found that one plant, it was just next to our building, was this Tragianulocrata. This is one of the endangered species, and it is having high medicinal activity. Okay, even the anti-cancer activity has been reported. And as it was endangered species, we could not collect too much amount of this plant. Best way is that you can go for the endophytic fungi. You isolate, and in the lab you keep maybe 10 conical flask around 500 ml, you will get enough amount of this endophytic fungi. So then I suggest maybe myself and my student, Dr. Anand, we discussed, we thought that this is the plant we are going to select. And the other plant we selected was that the Gingiber officinal. This is actually grown in that place a lot, that is especially Kushalnagar belt. This is the plant which is grown and it is marketed a lot. We thought that why can't we try even that one, because just next to our compound it was growing, because it is a farmland, and we could isolate nearly 15 to 20 endophytic fungi, and we tried to characterize it, maybe colony characterization and all. And later the problem is that which microorganism to be selected for further studies. Then we started doing the preliminary assay, and we selected some key microorganism which can be used for our studies. And what is the best way to go further? Because this is the costly business for us. At that time, at that situation, it was very difficult to characterize each and every microorganism, and it is a Herculean task. So what we did was that we selected few microorganisms, and we carried out the selected region where the DNA has maybe the RNA part has less maybe the changes. So conserved regions of the fungal region that was selected, and it was amplified, and we sequenced, and later on we carried out the analysis, and we related that one to the microorganism already reported in the literature. So we could find there are certain microorganisms, they can be related to these one. So these are the microorganism we could select, and we found that they have 99 similarity with the one which is already reported in the literature, so that we could select them as the species, strains will be different. So these are the different strains we have isolated, and these strains, we found that the strain which we isolated from Gingiber officinal, it is having actually not that much activity when compared to other extract or maybe the other fungi what we isolated from Tragianulocrata. So we studied only some few preliminary activities in this plant. So we got one paper and we left that one, because it is our intention was to go for the anti-cancer activity of the plant extract or maybe the endophytic enzyme. So what is the overall plan of our work? How did we work on it? We selected the plant, and this Tragea involucrata was selected based on the previous knowledge, and then we isolated the endophytic fungi, then we extracted the DNA, and that was amplified a certain region, ITS region we call it as. That region was amplified, and we constructed the phylogenetic tree to find out where exactly it belongs to the microorganism. And later on, we went for mass culturing this, the endophytic fungi. And once we mass cultured and we purified that compound, and this compound we came up with the, one of the compound, this is actually the one which is not reported earlier. We got a new compound, and that's why, that's what it took a lot of time. Here endophytic fungi, the major problem is that they are the tiny microorganism, and a small quantity of this secondary metabolites they are produced. So to get them itself, it is a Herculean task. And to go for this, the characterization and structure elucidation is another big task, because usually people, they will not go, if you look at any endophytic studies, they will take one extract and they will study all the parameters, they will study the LCMS detail, and they will conclude that this compound may be responsible for that particular activity. So our intention was not to go in that way. Instead of that, why we can't go for the bio-guided assay? So we studied the cytotoxic activity, angiogenesis activity, as well as anti-tumor activity with that particular compound. So usually how we do the study of efficacy of natural compounds. So the basic, what Dr. Nag usually used to tell, the MTT assay has become so normal, it is one of the small component of this study. But for us at that time, it was a big study to carry out because of our fund limitations and all other technical limitations. So we did that one, and later on even we studied not in the immunodeficient mice, we could not get that much, we couldn't afford that much, and we studied on the syngenic mouse model, and then we isolated and purified the compound, and then we studied again whether it is showing that compound what we have isolated, whether it is having anti-cancer effect or not. So this is the cytotoxic workflow. This you might be very familiar. This is a very simple assay. You will take the cells, you seed it on to the plates, and then you incubate at carbon dioxide incubator. Then once you add the drug, and once again you incubate in the incubator, and then you study by adding certain stains, or maybe like dyes, like MTT dye or XTT dye or MTS dye, or whatever it might be. And finally you measure the reading, and you will get what the result is. So we were going through that one, and we found that was a significant, and we have selected 4 endophytic fungi based on our previous knowledge. That's what I was telling, bio-guided assay. So we selected 4, and out of that one, this C2 extract, it was showing a better activity on almost all the cells. We selected one skin cancer cell, and the other one is that hepatocellular carcinoma cell, HepG2, and there also we found that this C2 is actually showing the better activity. The crude extract I am telling, it is at 1 mg itself, it was actually showing the activity to higher extent, because according to this NIH guidelines, the activity, if it is at one particular, say for example 30 microgram of this extract, the crude extract, it can be considered as a good drug to further study. We actually even studied on breast cancer cell line also. Why we selected this cancer cell line? Because the Tragea involucrata in the traditional report also, it is used in certain tribes in Kerala for treating the breast cancers. Based on that, we worked on the plant. We found that it is showing the anti-cancer activity. And then we wanted to know whether the endophytic fungal extract showing the anti-cancer activity on MCF cell lines. We found that it had a good anti-cancer activity on MCF, that too especially C2 extract, that is one of the one. And later we wanted to know what happens to the normal cell, because most of the occasion what happens when you work on the cancer cells, if they are toxic to the cancer cell, even they will be toxic the same extent on the normal cell also. So we wanted to find out what happens with this HEC cell. So we found that this C2 is actually less toxic when compared to the cancerous cells. We thought that this is the key fungi which you can focus on. So even we did different fractions of this endophytic fungi, and we studied the DNA fragmentation, whether it is having any effect on the DNA. We found that it is actually working at the DNA level, and when we treated the extract with the DNA, it is fragmenting the DNA into the fragments. That means it is maybe indirectly showing that it is causing maybe it is actually damaging the DNA and preventing the cancer. Then we went for the bioguided isolation of the purification of the lead molecule. So here the bioguided isolation is actually kind of a laborious work, where in each step we are checking whether it is acting as an anti-cancer, because our intention was to go with the pure compound. Okay, so we selected the plant, isolated the endophytic fungi, and then we had a limited facility. So what we did was that basic facility should be sufficient for us to go for the studies. That's why we selected two techniques. One, the TLC, and another one is the column chromatography. So those two were easy to handle and easy to carry out. So in the TLC, we could see different bands, and we went for preparative TLC, because preparative TLC is the one which is thin layer chromatography, where the layer is thick, so that you can scrap out the silica gel and you can take out the compound. So this is a laborious, but that is how we can get the compound easiest way, because you need not to lose the compound. And if you go for any other method, if it damages the structure of the compound, there is no novelty in, or maybe there is no maybe importance in isolating that particular drug. So then we went for once again purification, several cycles, and then we studied the different spectral parameters, and we took the help of the chemists. So we come up with the drug, or maybe the active component. This is the drug. Ultimately we got that one, and we could achieve nearly 87% of the purity. That is considered to be highest when in compared to the maybe any other reported literature. It is on par with many of the literatures where they are reporting about the purity of the drugs. So then we didn't stop there. We wanted to find out whether the what we considered as lead molecule, I don't call it as drug, it is a misnomer. So I call it as a lead molecule. So we wanted to check whether it acts on all the cell lines. Once again we repeated our study, and we checked on the A431 and HEC, MCF7 and HepG2. You can clearly see that on MCF7, the activity of this particular, we called it as F3, because it is having that 13 or 14% impurity. We called it as F3, that is fraction 3. And this fraction 3 had the high activity on this the MCF7 cells. And compared to other cells, the breast cancer cells was the one which is actually highly sensitive to this particular lead molecule. And then we checked with the non-cancerous cell line, that is HEC, and we found that it is not that much toxic when compared to the MCF7. So that means somewhere we actually could prove that whatever the lead we have isolated, it works on the cancer, but to lesser extent on the non-cancerous. So this later actually we wanted to work it out on the in vitro model also. So I will go a bit fast because I have to cover the time. So this we selected syngeneic model. We took the ascites carcinoma cells and we injected it into the mouse, and we studied. We got the details like this. It actually could it was not altering the tumor, maybe the mice weight, and even the these ascites cells were actually coming down when we treated with this F3 compound. And later on, as there is a link between this ascites tumor and angiogenesis, tight link, and the breast cancer also, the major that is key player is the angiogenesis process. That's what I was actually explaining, angiogenesis. So breast cancer and the angiogenesis, they are linked, and even the ascites tumor in the mouse is also, they are highly angiogenic. We wanted to find out whether it is acting as an anti-angiogenic. So briefly I will tell, this is the normal blood vessel, and once the cancer it develops, the cell starts dividing, they actually getting accumulated, and there won't be any the blood supply there. They actually become hypoxic in the sense they will have less oxygen condition, and this makes or induces the maybe the blood vessel to generate many more blood vessels, and it actually one of the hallmarks as I was telling. These are the different the approved anti-angiogenesis drugs, and we wanted to study what happens to the anti-angiogenic activity of the F3. So easiest way, because most of the things what we studied is the easiest way. Here actually Dr. Shankar from Davangere University, he was helpful in carrying out these studies. And here we carried out the chorioallantoin membrane assay, where we actually treated the hen's egg, and we found out that the number of blood vessels they are formed. So in the CAM assay also we could clearly see whenever we treated with the F3, there was a reduction in the angiogenic activity. And in the same way, even in the intraperitoneal membrane of the mouse, we observed that there is a less number of this the blood vessel formation, indicating that along with that the H&E staining, it is showing that it is actually preventing the blood vessel formation. And even we studied the cells by staining GMSA after treating with the standard drug cisplatin, F3, and the combination also we studied. We found that they are showing some apoptotic morphology, and indicating that that's why maybe it may induce the apoptosis, and it is actually acting as an anti-cancer. And we did not stop there, because the angiogenesis is closely linked with the one factor that is called the HIF1 alpha factor or hypoxia factor, hypoxia 1 alpha we call it as. So we studied that factor, maybe the expression or the amount of hypoxia factor that is maybe expressed during the cancer, and after treatment in the mouse, what happens with that one. We found that the HIF1 alpha, it is getting accumulated in the cytoplasm, but if you look at the nuclear fraction, clearly it is showing there is a reduction in the HIF1 alpha. HIF1 alpha. So that means here actually the thing what happens is that HIF1 alpha, it is present in the cytosolic region, and it requires or it has to bind to the beta, and then it enters into the nucleus. And there it will bind to the DNA at specific region, and there it will initiate maybe the expression of various downstream gene. They are responsible for the progression of the cancer. So our intention was that whether it is acting on the HIF1 alpha factor. We found that clearly it is actually preventing the translocation of the HIF1 alpha factor to the nucleus. So with our brief study or the basic study, we came up with a model. What kind of or what way our drug is acting. We found that this drug is specifically preventing the interaction between the HIF1 alpha and HIF1 beta, so that the HIF1 alpha is not translocated to the nucleus, and that's why once HIF1 alpha is inhibited, naturally the cancer maybe it is actually suppressing the cancer. So this is actually the normal the HIF1 pathway that let us not worry too much about. And along with that, we did not stop there also. We actually studied maybe the marine the endophytic fungi also, because not only this the terrestrial endophytic fungi, but at that time very few people actually they were working on the marine endophytic fungi. So I thought, why not we work on that? Because the marine is the condition where it is an extreme stress condition, and that's why whatever the compounds that are produced by the microorganisms, they are actually kind of they are produced in a different nick, so that they can work on the cancer in a better way. That's what our assumption was. And we could find that one, but only the thing was that we could not isolate the compound to that much extent when compared to this endophytic fungi, what we isolated. We reported many studies based on this, maybe the marine endophytic fungi also, but we could not isolate the active ingredient, that one. So instead of that, we studied other basic aspects, and we studied whether it helps in the formation of whether any other compound that we studied. And with this, I think I am crossing the time, actually 12:15 to 1:15, I think my time. So we can stop. I think you are also hungry. So if any questions, I am ready. Otherwise, we can have the questions later. I would like to thank my mentor, PhD mentor, Dr. Gopal Advi Rao, Davanagere University. Earlier it was in Coimbatore University. I thank Dr. Nag. He is actually instrumental in reaching Miami, and instrumental and making me to understand cancer biology in a better way and in depth way. And I also thank other collaborators. They helped us a lot to carry out this study, because without their help, I couldn't carry out this study. Dr. Santosh Kanade from Central University of Hyderabad, he was actually in the Central University of Kerala earlier. He helped a lot to carry out many of the cytotoxic studies and all. We had a very good collaboration, and even we could help to small extent his work also. And Dr. Shankar J, he is a good friend of mine. He helped me to carry out this the anti-angiogenic studies and all, and that's why we could reach the goal a bit faster. And Dr. Anu Appaya, he is the scientist at CFTRI Mysore. He helped us to maybe identify the microorganism, or wherever we are stuck, immediately we used to go to him. Sir, this is what happening. What is the next way to approach? He was very friendly. Actually, he was the faculty of Mangalore University, and he was a bit affectionate to the Coorg also. Regularly he used to come and he used to help us. And all this work, this laborious work, was done by the Dr. Anand. He took, because that time except test tubes and beakers, we could not think about anything else. Even power was not there at that time when we moved to the Chikkalavara. Only thing is that we were supposed to work with the limited, maybe the limited resources, but somehow we could work out that one. I thank my maybe the grant supporters, like UGC supported this work, sorry. And at the same time, I got a grant at that time, BRNS, for another study where we studied the radio protection studies. And ICMR is the one which helped me to reach Miami and learn pancreatic cancer, because earlier I never thought about, I will look at the pancreas. because of Nag, I could see the cancer of the pancreas as well as the normal pancreas. I used to think somewhere it is present in the stomach, it is nothing to do. But once I went, I could sense how the science works, because the basic science to the applied science, I saw a lot of difference in the science. That's why I wanted to share this information, because the way a scientist thinks and way a teacher thinks may be a lot of difference. That's why Nag, he might finding odd when I am presenting my details. It seems to be very preliminary. But the thing is that with my limitations and with my limited resource, or maybe that conditions made me to go ahead with that one. And even I had, recently I had a grant from SER on that one. Mr. Gagan, he is working, and it is an extended project of earlier what we did that we will be discussing tomorrow. And I also thank Ministry of Education. Actually, it supported this GAN program. Otherwise, it wouldn't have been possible. And even Mangalore University, because of Mangalore University, I could stand here and I could actually discuss my views. And I could contact Nag also because of Mangalore University. I thank Mangalore University also. And absolutely Nag will be appreciated for all the things that is happening now. With this, I conclude my brief work what we did. Thank you. Thank you. So if you don't have discussion, because we are running out of time, we can have lunch. And in the meantime, if you have any maybe the queries or any clarifications, we can discuss. 1:30.